Good morning, my name is Christine Piana, and this is a presentation on viruses. During this presentation, I'm going to talk about several aspects of viruses, including the characteristics of viruses that make them unique, the structure of viruses, the life cycle of viruses, how scientists and doctors detect viruses, how viruses are transmitted, common viruses affecting people, and the treatment and prevention of viruses that affect people. Unlike other organisms studied in microbiology, viruses are acellular, meaning that they are not made of cells. Viruses are also obligate intracellular parasites, meaning that they need a host to survive and replicate. A host range is the range of hosts that viruses are able to infect, because most viruses can only infect certain hosts, called host specificity, or they can only infect specific cells within that host, called host cell specificity. Viruses also contain very limited genetic material. They contain either DNA or RNA genomes, not both. A protein capsid surrounds this genetic material, called the genome. Viruses can also infect all organisms, including animals, people, plants, bacteria, and archaea. Viruses are also very, very small. They are typically between 20 nanometers and 900 nanometers. The chart on the right shows several organisms and their sizes. If you look at the flu virus, which is 100 nanometers, compared to the adult female, which is 1 meter, you can see exactly how small a flu virus is. There are 1 billion nanometers in 1 meter. So think about how small a virus is when it actually infects a person. There is an exception. There are new giant viruses. These giant viruses are really just a little bigger than bacteria, which if you look at the chart again, you can see that bacteria is not that much larger than the flu virus. So giant is really just a descriptive term because it's larger than typical viruses. The body of a virus is called a virion. That includes the nucleic acid, which is the DNA or RNA genetic material, surrounded by a protein capsid. That protein capsid is a protective covering over the genome. Protein capsids sometimes have an extra membrane surrounding them. Viruses with the extra protective coating are called enveloped viruses. The envelope is made up of part of the plasma membrane, which is made of phospholipids and proteins. If the virus protein capsid does not have an extra protective membrane, it is called non-enveloped or naked. Some viruses also have spikes that are little attachments at the end, as you can see in the top right-hand illustration here. These spikes aid in the virus's attachment to a host cell. Viruses also come in several different shapes. The first one mentioned here is the helical shape, which means that the virus is shaped like a cylinder. You can see an example of this in the bottom left-hand illustration of the tobacco mosaic virus. Another shape is called polyhedral, which you can see in the top left illustration of the adenovirus. Polyhedral viruses are 3D and they resemble a soccer ball. Other viruses are complex in shape, which means that they can have characteristics of both a helical and polyhedral shape. The top right-hand illustration of the bacteriophage shows the polyhedral head and a helical tail, and that is an example of a complex virus. Doctors and scientists use several different methods of classifying viruses. Doctors tend to use the International Classification of Diseases, which assigns a virus or other disease a number or a code. These numbers and codes are used to track the morbidity and mortality rates of viruses and other diseases affecting populations. The Baltimore classification is another method of classifying viruses. It classifies them into families based on the viral genome or viral replication mechanism. Other ways to classify viruses are by categorizing them based on whether they are naked or enveloped structures, single-stranded or double-stranded DNA or RNA, segmented or non-segmented genomes, positive strand or negative strand RNA, on their host specificity, tissue specificity, or capsid shape, and whether the viruses contain special genes or enzymes. The next topic I'm going to discuss is the life cycle of viruses. On this slide, you can see an illustration of the two methods of replication of viruses in prokaryotic hosts. On the left, we see the Lytic cycle, which has 5 stages, the first being attachment, when the virus attaches to the host cell, penetration, when the viral DNA penetrates the host cell, biosynthesis, when the virus replicates, maturation, when the new viruses are completely formed and ready to infect new cells, and the last stage being lysis, which is when the mature virus bursts out of the host cell and is free to infect new healthy cells. At the end of this lytic cycle, the host cell actually dies. The other method of replication in a prokaryotic host is the lysogenic cycle, which shares many of the same features. It begins with the virus attaching to the host cell and again penetrating that host cell. It then enters into the lysogeny stage, where the virus's DNA or RNA is incorporated into the host cell's genetic material. And then when that host cell replicates, it also replicates the viral DNA or RNA. The last stage of the lysogenic cycle is induction, where the viral genetic material is excised from the host cell, and it is then able to enter the lytic cycle, where it matures and bursts out of the cell and can infect other healthy cells within the host. At the end of the lysogenic cycle, the difference is that the host cell is not destroyed. The next aspect of viruses I'm going to discuss is their life cycles and how they replicate. When viruses invade prokaryotic hosts, there are two methods of replication, the lytic cycle and lysogenic cycle. The main difference between the two is that in the lytic cycle, the host cell dies, whereas in the lysogenic cycle, the virus's DNA is incorporated into the cell's DNA, but the cell does not die. In both cycles, the virus attaches to the host cell. It penetrates the host cell, meaning that the viral DNA is injected into the host cell. Then the virus replicates, matures, and in the lytic cycle, the cell bursts and releases the new virus into the host for it to infect other healthy cells. And in the lysogenic cell, the new viral information is excised from the host cell and released to infect other cells via the lytic cycle. When viruses invade animal hosts, they follow a slightly different cycle. The first two stages of attachment and penetration are largely the same. When it comes to the third stage, there is what they call uncoating, where the viral contents are released. It then enters biosynthesis, similar to the last life cycle we saw. And in biosynthesis, again, the genetic material is replicated. Next, it enters the assembly stage, where the new phage particles are assembled. And lastly, it enters the release stage, where new viral particles are released into the extracellular fluid of the host. And then from there, they are able to go on and find other healthy cells to infect. In contrast, when viruses invade animal hosts, they go through very similar stages, including attachment and penetration, but the next step, uncoating, is different because the viral contents are released in the cell. From there, the cell goes through biosynthesis and replicates. The new virus particles are then assembled, and then the virus is released into the extracellular fluid of the animal host, where it continues to replicate and infect the host further. When viruses infect plants, they are most similar to animal viruses. They can have a very narrow or broad host range, but most viruses that infect plants are transmitted by direct contact when two plants might be touching when they're growing, or by mechanical vectors, which I will discuss in the next slide. These viruses are also called biotrophic parasites because they can infect the plant without killing it. And another aspect of when viruses infect plant hosts is that for the virus to cause a systemic infection, which means that it affects the entire plant, the virus has to enter some part of the vascular system of the plant. Usually that occurs when there is some kind of opening on the plants, like a cut or some perforation, so that the virus can get into the vascular system. Doctors and scientists use many different methods to detect whether a virus is present in a host. One of the main methods is looking at cytopathic effects, which is really looking at the changes in cells. When a virus invades a host cell, the cells become diseased and they look different. So by looking at the cytopathic effects, one can determine whether a host cell is healthy, diseased, and if it is diseased, investigate the cause, which could be viral. Another method is polymerase chain reaction. And this test detects the presence of viral DNA in a tissue or blood sample by making many copies of the viral DNA and then using nucleotide sequences to see if they bind to the specific sequences of the viral DNA. And by looking at this binding of the nucleotide and DNA sequences, scientists can identify the virus. An electron microscope is a tool used to view viruses. Electron microscopes use a beam of electrons to create an image. These microscopes have a much higher magnitude and much higher resolution than regular microscopes. Because viruses are so small, the electron microscope is necessary to view them. Another test used is blood testing. A blood sample can be tested for viral antibodies or antigens. And lastly, another test is a serological assay, and a hemagglutination assay is a specific example. And this is a test done where a sample of the virus is isolated in the serum, which is the liquid portion of blood plasma. So we have an isolated portion of the virus, and then red blood cells are added to that isolated sample, and if clumping does or does not occur, could indicate the presence of a specific virus. The next aspect of viruses I want to discuss is how they are transmitted. They can be transmitted in many different ways. One is through direct contact, and that can be one person carrying the flu virus shaking another healthy person's hand and it being transferred from the surface of the infected hand to the healthy hand. They can be transmitted through indirect contact with a fomite, and a fomite is a non-living object that carries a virus. So if a virus is attached to a kitchen counter or a doorknob or something of that nature and it is picked up by another person, that would be in direct contact with a fomite. Viruses can also be transmitted via vectors, and there are mechanical vectors and biological vectors. Mechanical vectors carry the virus on the outside of their body. So, for instance, if a bird were carrying a virus on the outside of its body and it came in direct contact with another animal or another host and infected that healthy host, that would be an example of a mechanical vector. Biological vectors carry the virus on the inside of their bodies. Typical biological vectors are like flies, ticks, mosquitoes, where the virus is inside of them and they bite a host and transfer the virus to that host. Another way that viruses can be transmitted are via contaminated food or water. Now that we've talked about the basics about viruses, I just wanted to talk about a couple of the common viruses that affect people, including the common cold, the flu, chickenpox, human papillomavirus, hepatitis, and HIV and AIDS. All of these are very commonplace in our society, and they are all caused by viruses. To the right, I have images of couple viruses that affect the skin in people. So you can see how different viruses manifest differently. The patterns of the breakout and lesions from each varies, and this is a good indicator of the cytopathic effects method of identifying a virus. HIV and AIDS, which I mentioned as a common human virus, is actually called a retrovirus. Retroviruses contain special enzymes called reverse transcriptase. And in these cases, the viral DNA becomes a permanent part of the host chromosome. Luckily, today, we can treat HIV and AIDS using antiviral medication. However, that was not the case until the last couple decades. Unlike AIDS and HIV, which can only be managed and controlled, not completely eradicated from the body, many other commonplace viruses can be either prevented and or treated. Vaccinations are available for such things as the flu, chickenpox, and HPV, which is one of the most common sexually transmitted infections in the United States today. Vaccinations are very helpful in preventing viruses, in particular strains of viruses, from infecting humans. We can also take prophylactic measures, especially in the winter and back to school when viruses are running rampant. We can wash hands and avoid other sick people and use disinfectants for these commonplace viruses. When it comes to sexually transmitted infections caused by viruses, using condoms and limiting sexual partners and receiving yearly checkups can definitely help limit or at least identify whether a virus has been transmitted to you. There are also medications available. Some can treat the symptoms of viruses, and others are actually antivirals that treat the virus itself. This concludes my presentation on viruses. We've talked about the fact that viruses are acellular and cannot survive or replicate without a host, that viruses come in several shapes and are extremely small, that the life cycle of viruses depends on the host that it infects, that scientists and doctors employ several methods to detect viruses, that viruses can be transmitted by direct or indirect contact, and that many viruses affect people, but most can be prevented through vaccines or treated with medication. I hope this presentation has given you a brief overview on the ins and outs of viruses and will be helpful learning more.